Um, Hello, everyone, and welcome to today's webinar. It is my pleasure to introduce our speaker for today, Professor Shilpa Chokshi. And she's Professor of Experimental Hepatology and Environmental Hepatology at the University of Plymouth. And if I'm not mistaken, I think that she's also affiliated with King's College London, and she also serves as the Acting Director and Chief Scientific Officer for the Roger Williams Institute of Hepatology. Professor Chokshi is a leading translational scientist in chronic liver disease research, and her expertise is spanning from viral hepatitis to cirrhosis to liver cancer. And her work focuses also on understanding the molecular mechanisms or mechanisms in general of liver injury and repair, with particular focus on environmental toxins and plastic pollution, which we'll hear also about today. So specifically microplastics and nanoplastics as well. And her work is also focused on identifying novel drug targets and biomarkers, so addressing in this way the complex challenges of treating liver disease. In her multidisciplinary research, she also aims to translate these scientific discoveries into more meaningful and patient-centered interventions, and also to advance public health awareness. So before we begin, just a quick note for everyone, we will have a Q&A session at the end of the presentation, but feel free to drop any questions at any time during the talk if you have something on your mind, and then using the chat box, then we will address everything at the end of the presentation. And now I think I'll hand things over to Professor Chokshi. She will present her work entitled Micro and Nanoplastic Pollution and Liver Disease, an Emerging Health Concern. I think you should be able now to share your screen, and yeah, you can go ahead. You can start your presentation whenever you're ready.
Yes, thank you very much for the kind introduction. And thank you very much for giving me the opportunity to discuss with you this emerging health concern. And I'm going to start with a little bit of a caveat to say that this is emerging. We're still waiting for mechanistic data, but I hope to convince you that this is something that anybody that's working with patients with liver disease should be aware of. So, I don't think I need to introduce this to this audience, but it's nice to start off with this slide, which is that we are living in the middle of a global liver disease epidemic, and the three pillars of disease globally are viral hepatitis, obesity, and alcohol. And of course, obesity and alcohol seem to be the main drivers in the Western world. And these drivers of injury are promoting this huge burden of cirrhosis, the 11th commonest cause of disease now, which is driving these projected rises, exponential rises in liver cancer. And actually, liver disease in Europe is particularly important, and I still count myself as European, even though I live in the UK. It is that liver disease remains one of the highest contributors to mortality rates, with huge numbers of deaths per year, with ALD and MASLD accounting for the majority of the burden. Of course, MASLD, as we all know now, contributes to about 30% of disease in patients, higher in those with other susceptibilities. And alcohol consumption, actually in Europe, we have the highest prevalence of binge drinking and the lowest rates of alcohol abstention. But these primary risk factors, these injuries, do not actually fully explain the risk or the liver disease severity. And in fact, there's been a number of different studies and editorials, perspectives showing that actually, whilst excessive alcohol intake is directly linked to the severity of liver injury, only approximately 30 to 40% of heavy drinkers actually develop severe alcohol-related liver disease. So alcohol doesn't actually fully explain the risk. This is also true for MASLD. Only about 20% of individuals with hepatic steatosis progress. So metabolic risk doesn't alone explain disease progression. And I think, we're all very, very comfortable with this hypothesis of the multiple hits. So there are other modifiers such as the microbiome, sex, genetics that can accelerate injury on a primed hepatic background. And these additional hits can actually amplify injury, inflammation, fibrogenesis, promoting disease progression and severity. And the new kid on the block is, of course, environmental pollutants. And the evidence implicating pollutants as a tangible, modifiable risk factor for MASLD and chronic liver disease is actually quite striking. And there are papers showing that pollution, other exposures, even occupational exposure and microplastics are actually directly correlated with this exponential increase in SLD that we have seen. And actually, if you look at the data, you can see here, I've just plotted the global plastic waste since the 80s and liver mortality rates. And actually, if you put them together, they look very paralleled. But is this just correlation or is it actually causal? And that is the work that I moved from King's to the University of Plymouth to do. So what I want to know is there a link between plastic pollution and liver disease? So plastic pollution, actually, it's a giant problem. Everywhere you look now, especially with my rose-tinted glasses, is plastic. So over 400 million tons of plastic waste is produced per year. This is predicted to triple, if not quadruple, by 2060. And about 9% is recycled. And these plastics hang around forever, and they exist as micro and nanoplastics in the open environment. And actually, here's a nice infographic that you can find, that this is the amount of global plastic waste per year. And on the left-hand side, you can see, for scale, the Burj Khalifa. And the majority of this is single-use waste plastic. So what are micro nanoplastics? Well, actually, there are primary microplastics, MPs, which actually are directly released into the environment. These are mainly from air pollution, particulate matter, textiles, industrial treatments, personal care products, glitter, for example. And actually, if you look in the ocean at what types of plastics are out there, the majority are from synthetic textiles and tires. Now, over time and through degradation and fragmentation, because of oxidation, mechanical forces, ages, these plastics degrade into secondary microplastics. So you get this production of these secondary microplastics, and further degradation, fragmentation, produces nanoplastics. And you can see the kind of size limits that we're kind of talking about. So microplastics, particularly at the lower end, are thought to be found in tissues, and I'll show you some data later. Nanoplastics, actually, they're small enough to get into cells, perhaps even organelles. So there's a little size reference for you. You're talking about DNA virus-sized plastics. And actually, if you look at how they're getting into the human body, and I'll talk a lot more about this. Of course, these plastics are found in the air that we breathe, the water that we drink, and the food that we eat. Actually, nanoplastics and microplastics are in the respirable, inhalable spectrum. And whilst the analytical toolkit for detecting these plastics is getting better, we're just at the beginning of this technology. And actually, at the smaller scale, we find it very difficult to detect these plastics accurately. So, I mean, at the lowest scale, we're talking about the width of a strand of DNA. And to try and visualize this and to measure this and characterize this is still extremely challenging for us. And I think some of the discrepancies that we see in the literature, and there's been a lot of press about this, may be because it's not that we don't have plastics in our bodies, it's because we can't accurately say it's this burden. So what about the history? Well, mass production of plastics only began in the 1950s, if you can believe. Whereas if you look around you, there are plastics everywhere now. They started to be seen in large quantities in the seawater around the 70s. And in 2004, my very good collaborator from University of Plymouth, Richard Thompson, in 2004, actually published the first microplastic paper. And since then, we've found them in a number of different sources: drinking water, outdoor air, human stool. And more recently, we're now beginning to detect it in almost every human system and organ. This was a paper that Richard Thompson did 20 years after his first paper, basically showing that micro nanoplastics are ubiquitous environmental pollutants, and you can find them from the deepest oceans here to the highest mountains. And they're found throughout the entire natural habitat and in species that reside in this habitat, whether they be ocean species, whether they be birds, but in the natural environment, you can now find it in these animals, birds, and aquatic organisms. And this is a very nice infographic as well from the Center of International Environmental Law, suggesting that we actually probably inhale over a year the height of 2 giraffes worth of plastics, and over a lifetime, the height of the Eiffel Tower at a minimum. Whether this is true or not still needs to be really quantified better in large population groups, but the emerging evidence is pretty striking. Now, everywhere there's been a lot of, let's say, hype about microplastics. You could be swallowing a credit card's worth of plastic every week. Do I think that's true? Probably not. But I think at the extreme ends where you have environmental or occupational exposure, it's possible. Again, remember the detection techniques still need to be advanced, but they're everywhere now. It's on Netflix, it's in our big papers. We find plastics everywhere. And actually, this study very nicely summarized that you can find plastics, microplastics in the hair, saliva, sputum, blood, breast milk, the liver, kidneys, colon, placenta, the meconium, and in stool samples. I mean, plastics are ubiquitous within the human body and in the brain. Now, in terms of liver exposure, the primary exposure route is probably via the GI tract, so whatever you eat or drink, passing through the gut and then being filtered or through the liver, through the portal vein. But also, the majority of what is being inhaled will also end up in the GI tract, because whatever these particles, particulate matter, are deposited in the upper airways and they're cleared via the mucociliary escalator. So you get this secondary GI ingestion. Whether dermal absorption is important in hepatic micro and nanoplastic exposure is yet to be determined. And these microplastics in these organs are now being associated with disease. So there's higher microplastic burden associated with dementia. Microplastics found in the cerebral spinal fluid are associated with an aneurysm risk, with Parkinson's disease, and major cardiovascular events, such as myocardial infarction, stroke, and death. And they have been reported in cirrhosis, which I'll talk about later, and associated with MASLD. But these are all association studies. They're not necessarily causal. And sometimes one of the criticisms currently is that, well, phone use has gone up, but is that associated with liver disease? And that's something that we're now currently trying to ask. And in terms of the liver, well, there are papers that are emerging now showing that actually, environmental pollution is a tangible risk, that micro and nanoplastics can act as obesogens. There was a very nice study in Journal of Hepatology recently showing an increased risk of MASLD in areas of high pollution. And this recent editorial asked whether hepatologists should care about air pollution. And from what I have seen and read over the last year of starting the Center of Environmental Hepatology, I think the answer is a resounding yes. So, are they relevant to a disease in the liver, ALD and MASLD? Well, we showed previously from my group that actually, if we talk about alcohol-related liver disease and cirrhosis, that these were really 3 primary drivers of disease: inflammation, gut dysbiosis, barrier failure, hyperpermeability, endotoxemia, and a susceptibility to antibiotic-resistant infections. And actually, all of these 3 pillars have actually been found associated with micro and nanoplastics. And there is a wealth of data from animal models that micro and nanoplastic exposure in animal models, predominantly mice, drives gut microbiome dysbiosis, gut barrier damage, and immune activation locally in the gut. Peripherally, endotoxemia is associated with micro nanoplastic exposure. There's accumulation in the brain, kidney, and liver, and it drives systemic inflammation. And there is this evidence that hepatic accumulation is associated with disease. And this was a study in 2022 where they found 6 types of microplastic polymers associated with liver cirrhosis, but not in those with underlying disease. And this paper actually, whilst it was groundbreaking, probably underestimated the true disease burden because of this detection issue and this lack of ability then to detect trace amounts of nanoplastics. And future studies are really important to evaluate whether hepatic microplastic accumulation represents a potential driver in fibrosis or whether it's a consequence of cirrhosis, the leaky gut, and this increased translocation of content from the gut to the liver. Which is it? Is it chicken or is it egg? And that data still needs to be shown. This is another study from New Mexico from Matt Campen's group. And he did a post-mortem study assessing organ accumulation of micro nanoplastics in 28 cadavers using a more sensitive detection method. And they showed that the brain, kidney, and liver showed significant microplastic accumulation, and it wasn't influenced by age, sex, ethnicity, or cause of death. And what was really interesting was hepatic microplastic burden significantly increased between 2016 and 2024. So it was this huge increase. And is this of concern? Well, according to this study, it's the equivalent of a paper clip's worth of plastic in the liver. But I want you to see this lens through the fact that there is plastic in the liver, but is it exactly that amount is something that needs to be shown in larger studies. So this changing plastic fingerprint in the liver is really quite enticing to understand. You can see here the breakdown of the type of plastic polymers that were found in the liver in 2016 and then in 2024. So you can see a huge increase in PVC, in nylon, in styrene butadiene rubber, which is what tires, car tires are made of, and in polypropylene. Personally, I think this reflects a lifestyle shift in the last decade. We know that plastic production is booming, there's rising pollution rates, and everyone's really into this fast fashion. And with this e-commerce growth, you think it, you buy it, and it turns up in 25 different plastic bags from online shopping. We also have had a real dietary shift. We had this increased reliance on processed food. And let's not forget, post-COVID, there was this healthcare surge, IV bags, PPE, gloves, syringes. Also, the way we live has changed in terms of synthetic flooring, furnishings. And of course, let's not forget, the plastics that are in the environment are fragmenting. So over time, there is a bigger bigger burden of micro nanoplastics for us to eat, ingest, and inhale. So I just wanted to highlight that actually the evidence in steatotic liver disease is actually really quite compelling. I mean, there's been 3 papers, I think, in JHEP, but these studies show that actually environment, pollution, and liver disease are intimately associated. We are what we eat, and the environment is directly affecting this disease, which is going to be hopefully classed as non-communicable. So it is a disease of our environment, of our exposome. So why would environment not have an impact? And I think it really underscores this need to understand these drivers together in the context of alcohol, fat, and perhaps even viral disease. As I'll show you later, these plastics harbor viruses, bacteria, and fungi. So I wanted to pick out some of the rodent data that I found particularly interesting. So this is a study where they gave mice models microplastics. These were polystyrene with high-fat diet. And what they saw was increased steatosis, inflammatory cell infiltration, ballooning, and a significant exhaustion of T cells, which we know are critical for tumor immunosurveillance, with exacerbated pro-inflammatory responses. This is relevant to alcohol-related liver disease, MET-ALD, not just MASLD. one of the criticisms of these studies, and I would be the first to say this, is that the plastics tend to be used in animal models, one. Two, they tend to be polystyrene for some bizarre reason. And three, they do not represent environmental plastics, which are multi-polymer, multi-size, and we need to be more aware of this. And a lot of these murine studies are using doses that are super physiological. So again, we need to be very mindful of the data that we are generating in the future. However, the data does show that there is this increase in disease. My slides. Okay, there we go. This is another study. I just wanted to highlight human food sources such as chickens. Actually, if you give chickens microplastics, and we know there's lots of microplastics in soil because of constant erosion from the air and from the water. But these microplastics actually have a huge effect on these chickens. There are changes in the gut microbiome, there are changes in their growth, in the liver and the kidneys, as inflammation actually acts as antioxidants. And this is what we're consuming. And so this is what is possible going to be happening to us, this type of metabolic dysfunction. Now, these plastic particles are more than just plastic particles. Actually, they bring other things along with them. They actually become colonized by different microbes very quickly because they're ideal attachment sites for bacteria, virus, fungi, because their surface chemistry can enhance microbial adhesion, and they act as little shields and pockets from UV and desiccation. And there is quite a lot of data now showing that there is an enrichment of antimicrobial resistant genes and MDROs on micro nanoplastics because they tend to enhance horizontal gene transfer because they're close localization on these plastics. And of course, these are highly relevant for our patients with cirrhosis, for alcohol-related liver disease. And in this review by Yang et al. Recently, it was, they did a review of all the data out there on how microplastics are remodeling the gut microbiome. And actually, there are associations in infants, preschool, children, and adults that microwavable meals and ultra-processed foods, because heating plastics releases microplastics and nanoplastics. So there is this association with stool concentrations. And there is an association with colorectal cancers, inflammatory bowel disease, again, highly relevant to our patients along the gut-liver axis in patients with cirrhosis. And actually, size matters because if microplastics are degraded into nanoplastics, well, they might become more bioavailable, more toxic, and also increase the absorption of contaminants, such as antibiotics, which we think is another driver of antimicrobial resistance on these particles. Moreover, I think what is really concerning to me is the number of plastic additives in these micro nanoplastics. So there are about over 30,000 plastic additives. A lot of them have not had toxicity studies. Most of them are not listed on the plastics that you buy, even if your food comes in that plastic or your drinks. They are flame retardants, plasticizers. About 10,000 of them are known to have endocrine disrupting, carcinogenic, and obesogenic qualities and driving these kinds of diseases. And these particularly interest are phthalates and bisphenols, which, again, early data is now showing that they're driving SLD inflammation. There's also data now showing from animal models that microplastics induce multi-organ toxicity. And if we think about acute on chronic liver failure and these second hits of disease, then I think this is very relevant. How these plastics move throughout the body is something of real interest. Are they packaged? Are they in lipid coronas, which allows them to move in and out of cells? Because not only do they have microbial coronas, but they also have protein coronas, lipid coronas, depending on where they are sitting, whether they're sitting in the gut, whether they're sitting in adipose tissue. But we do know that there is this multi-organ toxicity with some shared mechanisms of oxidative stress, dysregulated cell death, particularly pyroptosis, mitochondrial dysfunction, and inflammatory response. And I think that there is now enough mounting evidence to suggest that plastic-induced liver injury is something that we need to be working on. It's not just the plastics, it's also the co-pollutant chemicals, but we're talking about things that are very relevant to what we already know about disease, liver disease, gut dysbiosis, intestinal inflammation, systemic inflammation, fibrogenesis, mitochondrial damage, and cell death. But the mechanistic data is still not there. So I encourage you all to come and work in environmental hepatology, because this really is something that we need to become more aware of. So some data, there's no direct data, but there's some indirect data that this may be important. And I have a historical interest in alcohol-related liver disease, so I just wanted to show some of this data to this audience. So there was this study done in 2022, small study, not by hepatologists, and most of the studies are not done by hepatologists. They're done by environmental chemists, and they looked at, burden of microplastics in beers, mineral water, and tea. And actually, some of the beers, glass bottled beers, had very high amounts of microplastics, approximately 55,000 microplastics per pint. The predominant polymer type was polystyrene and polypropylene. And you remember polypropylene was the one that was on the increase from 2014. And beer packaging was not the contamination source because these were glass bottles. So they think it's the raw ingredients, the hops, the things that go into beer that actually are driving these large numbers of microplastics. And interesting, there's a person in my group at the moment who recently published a paper showing that vegetables have very high levels of microplastics. And he was working on radishes. But again, these raw ingredients have levels of microplastics that are then going down into the food chain and then turning up in us. So I think for chronic beer consumption, I think there is a microplastic exposure risk. But has anybody looked in a large number of beers in cider, which we know that patients with severe alcohol-related liver disease are drinking because it's cheap and they're all in plastic bottles, but nobody's really looked. If you look at wine, so I don't think wine drinkers are going to get away from this. Actually, when they changed the stoppers from cork to plastic, there seems to be a tangible risk. So synthetic polyethylene stoppers is a microplastic source. And most bottles, 26 out of 26 in this study, showed high levels of polyethylene. So for individuals with regular wine intake, this is a credible, cumulative, and quantifiable microplastic exposure risk. And again, other ultra-processed food, microwaving, these are all additional risk factors that patients perhaps with MET-ALD or MASLD additionally have, apart from the endocrine disrupting additives and chemicals that are in these plastics. I also wanted to touch again on some of these chemicals, and I think you've probably heard of PFAS or these forever chemicals that persist for years. The half-life clearance in humans is 6.2 years. And one particular interesting one is perfluorooctane sulfonate. And in this study, actually they showed in a mouse model of chronic binge ethanol feeding that PFOS aggravated liver injury, and that the liver was a dominant PFOS sink during co-exposure. So 60% of the administered PFOS actually accumulated in the liver, possibly for 6 years. I'm not sure the animals will survive that long, but it does position PFOS and ALD as an ALD risk modifier. And there was this paper last year that showed that actually gut microbiota can also bioaccumulate and hold on to this PFAS. So what is the consequence of that on liver disease? Well, currently that remains unknown. So I think the umbrella term of environmental hepatology is something that we should be working in and should be very aware of. The data is there that there is associations, but causality, well, I think so. There are a number of things that we need to get over and to advance to be able to make these associations. First of all, there's no legal definitions of the size of micro and nanoplastics. They are complex. They're not single polymers. They're not single size. They're not all spherical. So we need to really, the work needs to, recapitulate that complexity. Many different materials. Apples are not pears. You can't work on polystyrene and assume that's true for all polymers. There still remains no reference material. We have very little ability to detect the very small sizes of nanoplastics, which I think are probably causing the most toxicity. Most people are using pristine particles, whereas we think we need to be using aged particles, environmentally relevant particles. So there's quite a lot of work to be done. However, that still doesn't undermine the fact that the emerging data is suggesting that this plastics that we are inhaling or ingesting and that end up in the GI tract, translocating in the gut, probably at higher levels in patients with cirrhosis because of leaky gut. And if you look at the impact of microplastics on immune cells, hepatic cells, and mitochondria, then you can actually see individually on a cellular basis, they are driving disease pathways. The microbial corona that is picked up through the gut on these plastics that we ingest is likely to be hugely important in also driving disease and infection. So what we're now doing at the center that I've established in Plymouth, and the reason I established it in Plymouth is because of Richard Thompson and his work in microplastics in the oceans, allowing us a real base to be able to do this work. And we are now using patient-derived organoids, precision-cut liver slices, using environmental plastics that we are now cryomilling in the lab to understand with these co-modifiers what the pathophysiological mechanisms of plastic-induced liver injury are, and also if there are any mitigation strategies. So our objective is really to establish definitive evidence-based insights on the impact of MNPs on liver health, and really to develop ways to try and reduce not just hepatic, but systemic burden of disease. As I said, we're using environmental relevant plastics. We are actually inducing plastosphere biofilm formation, and we're looking at this together with alcohol and fat on lots of new approach models. Here's some preliminary data from the center where we've only been open for about a year, but I wanted to show you some data currently. These are immortalized human hepatocytes exposed to polystyrene, nanoplastics, fluorescent. And what you can see is as you increase the concentration, the actual cytoplasmic accumulation of these plastics increases concerningly. If you take precision cut liver slices, you expose them to a whole load of different polymers. What you see is actually some plastics seem to be worse than others. And here is ALT levels, AST levels of polypropylene, and you can see really high levels of AST, ALT from these human precision cut liver slices. So they do seem to be inducing some kind of injury, and about 93% of the cells show accumulation after 48 hours. Interestingly for us, if you actually expose these nanoplastics to bodily fluids beforehand, trying to recapitulate what happens in the body, you actually get even quicker rates of accumulation and much higher rates of toxicity. So serum conditioned plastics markedly exacerbate toxicity. We think it's because, well, we know because we've been looking, we think it's because they become fragmented even further. They're also producing leachates and chemicals, so the chemicals are leaching out. And in some of the precision cut liver slices, they were from patients that had alcohol use disorder. And in those patients, we see the highest amount of necrosis after exposure at concentrations that we know are found in liver from the studies that I showed you earlier. This is some EM. This is an immortalized human hepatocyte exposed to nanoplastics. And you can see here that the plastics are accumulating within the cytoplasm, and the cell has effectively become a plastic landfill. And if you look at higher magnification, you can see that they are in endosomes, probably because this cell is trying to get rid of this toxin. But actually, they become ruptured as they become full, and they're then released into the cytoplasm. We're currently doing RNA-seq and some laser dissection to understand the effect on that cell, and single cell sequencing, so we'll know a little bit more. When we show the precision cut liver slices to the histologist, a histopathologist, blindly, I mean, they think it looks like dilly, but that is just early observation. So I want to reach out to all of you and say, come and have a look at the work that we're doing. Here is the website. And, be aware that these patients that you see and the work that we're doing is hugely impacted by microplastics, by chemical additives, by plasticizers, and by pollution. We're working on a number of different studies. We are trying to improve the visualization and nanoscale characterization in tissues, especially the gut and the liver, and in immune cells and the blood as well. We are trying to advance these techniques for environmental monitoring. We're very interested in the biofilm, particularly the microbial biofilm, and how that drives hepatic inflammation. We have some clinical studies that we're doing to understand the impact of IV or in critical care on, let's say, levels of plastics in the blood and systemic circulation, and also the effect of microplastic ingestion. And we're trying to define the mechanistic pathways associated with plastic-induced liver injury. So I'd really like to thank the center, everyone in the center that's helped me establish this and the work that we're doing, particularly Dr. Paula Barrera, Professor Richard Thompson, fantastic collaborator and inspiration to us, and my previous team at King's and all the work that I did with them before I left to pursue this very exciting area of research. I'd really like to thank you again for the invitation, and I'd be happy to take any questions.
Thank you very much for your talk. Very inspiring, very